The relation between the gene and the DNA can be very well studied by mutation studies. Now, you know what is gene, you know what is DNA, you have an idea about it, though you don't know what it means in great depths, but you don't know what mutation is. Mutation is a sudden change, okay, an undesirable change, a sudden change at the molecular level. Now, when we talk about molecular level, and as I told in the beginning that if we have to study the relation between a gene and DNA, we better do mutation studies. That means it would come and affect the DNA directly, and that effect would be termed as mutation. Now, talking in molecular terms, mutation basically is any undesirable and sudden change in the DNA. Okay. Now, the DNA has to be affected. Now, recall the biomolecular structure of DNA. What the biomolecule of DNA looked like? It was nothing but it was a polynucleotide, right? Now, this polynucleotide is identified, or as I told you, this DNA could be studied in terms of gene as well. We can study gene or DNA. The gene has an expression and the gene is made up of DNA, and the DNA is made up of polynucleotides, which are further made up of base pair arrangements, right? Base pairs are there. There is sugar, there is phosphate. This is what we have. So, if I am talking about a gene, the gene is going to give some expression. Supposedly, there is some sudden change at that level that brings the expression of this gene totally changed, okay, then that is known as mutation. Now, this gene's effect will be altered, that means mutation is working. Now, how this gene has been affected? It has been affected because the DNA composition has been altered. Now, what is DNA made up of? Base pair, sugars and phosphates. Now, supposedly we have a DNA sequence like this. It is having A, T, G, G, C, A, T, T, A. Right. Now, it will have complementary T, A, C, 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 G, T, T, A, T. Now, supposedly the mutation has to occur. What will happen is that this sequence will change, or this entire codon triplet would change, or this would be replaced. So, when the change, undesirable change takes place at this level where the base pair chemistry is altered, alright, that is known as mutation. Now, the mutations are of two types. One is spontaneous and other is induced. As the name suggests, one would be occurring naturally, okay, at the biomolecular level, and one would be that type of mutation which would be brought about deliberately by subjecting the genetic material or the cell to some sort of mutagen. Mutagen is an agent that brings about mutation. So, the mutagen brings about mutation. It could bring about induced mutation if you are choosing the mutagen and then bringing about the mutation, and if the mutagen is present naturally and that brings about the mutation, that is known as spontaneous. Now, these mutations are the central character in evolution as well, as it was proposed by Hugo de Vries that the mutations, when they act as saltations, they work as the driving force for evolution. So, we have these two types of mutations. One is induced. You use certain mutagens like gamma rays, beta rays, you have X rays and other radioactive substances which would act and induce a mutation. Certainly, it would be at the genetic level, it would be altering the base pair composition of a gene, and it would be done deliberately. Now, supposedly the spontaneous mutation is occurring. How do we study spontaneous mutation? What are the two different types of spontaneous mutation? The two different types of spontaneous mutation are substitution and frame shift. All right. Now you know a codon is responsible for expression of gene. I believe you don't know. We haven't done it till now. If you're watching the lessons in series, you must not be knowing what it is. But you know that the DNA is arranged in such a manner that a fixed arrangement of nucleotides is going to code for a gene. Now we are going to take an example. Supposedly, we have an arrangement where we have the cat ate the rat. Supposedly what happens is that in place of C, I put R. So you see the entire meaning of sentence is changing, right? In the same way, supposedly I have, in the case of frame shift mutation, the cat ate the rat. Here what is done, that something comes and it pushes the entire frame. Now supposedly I put another E over here. Okay. So what will happen? Now the entire frame will change. The cat ate the rat. Now nothing has happened, only thing is one substitution has come over here and the entire frame has shifted. This is the example of frame shift mutation, and this is substitution when at place of one particular the particular alphabet has been replaced by another alphabet. In both the cases the entire meaning of the sentence is changing, but you can very well figure out that here the sentence remains the same. If you have to read it, the cat ate the rat, but the composition has changed, and this altogether is going to change what is to be formed. In this case also the outcome would be different. Now in this case of substitution mutation, we have two types of mutations. One is transition, other is transversion. Supposedly purine is replaced by purine. We have this arrangement A, T, G, 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 C, C, A, T, and this would be T, A. This is your DNA sequence, that is the gene. Now supposedly this G got replaced by C. So in order to maintain the complementarity, this comes G over here, it is altogether changed. The meaning would be changed. This is known as point mutation. One classic example of point mutation is sickle cell anemia, where GAG is replaced by GUG. This single change from A to U, it in terms of amino acid sequence I am talking, it replaces glutamic acid at place of valine, and that is responsible for giving sickle cell shaped to the RBC. So you see, one point change is bringing so much change in the entire body composition. So is in the case of substitution, but in one case purine is replaced by purine, another case pyrimidine is replaced by pyrimidine. Then in that case we have transversion. So you can make it out that if the purine replaces the purine, that would be supposedly at place of A comes G, right? Then it would be transition, but in case it happens to be C and in place it comes T, then the pyrimidine is replaced being by pyrimidine, then the condition is known as transversion. Okay, so this you have to keep in mind. Now in case of frame shift, if we see it in terms of for the triplets, which would further code for amino acids you will study, then in case the frame is one point is being added or being removed, then the entire meaning of the sentence is changing, then the entire further frame gets shifted and it gets changed. I would give you another example. Ram had red cap. Now this frame shift could be of two types. One would be insertion and other would be deletion. Alright. Now supposedly you add over here A. Now it would go like this. You see, after A the entire frame has changed, before it it's okay. Now supposedly there is deletion. Ram had red cap. There is deletion. This R goes from here. Then what it would be? It would become EDC. It would become APA, something like that, and forward. So as this deletion has taken place from here, so the forward frame has changed. Addition has taken place over here, so the forward frame has changed, and this is known as frame shift mutation, which could be studied in terms of insertion and deletion. We have substitution, which could be studied in terms of point mutation, and we had the example of sickle cell anemia, and this is all you have to keep in mind when you study about mutation at the molecular level. This mutation has lot of importance in terms of evolution that we study when we do evolutionary studies, but for now this much is